Hello, my curious minds, and grüß euch, as we would say in Austria. Welcome back to the HEAS podcast. I'm your host, Victoria, and I'm happy to be back with more exciting science knowledge and 3 new guests. This time, our scientific spectrum, if you will, is very diverse. We have 3 guests from 3 different fields: radiocarbon research, archaeology, and population genetics. My first guest is Mike D. He's the head of a radiocarbon dating facility in the Netherlands, and we chat about the first evidence of Vikings visiting the North American continent, interestingly predating the official discovery of America in 1492. We chat about radioactive bomb testing, which led to a sudden increase of radiocarbon in the atmosphere, and its current use in science. And we chat about why Mike thinks being a decent human in science is important. Probably not only in science, but overall. Our second guest is Mattias Jakobsson, who's doing research in the field of population genetics and human evolution. His lab is using very cool computational approaches to uncover ancient human migrations and human genetic variation. We talked about early Neolithic farmers and their migration into Europe and Africa, and the genetic differences as well as similarities of human populations. Mattias also explains which challenges he faces when studying the genomes of these ancient human populations. And last but not least, our third guest is Wil Roebroeks. He's an archaeologist with years of experience and many great stories to tell. Although officially retired, I don't see Will settling down and abstaining from science anytime soon. He talks about years of excavation at a huge German brown coal mine, and that they found a whole Paleolithic zoo of bones, including those of mammoths. We also discussed the use of red ochre in Neanderthal burial contexts and the implications of this complex ritualistic behavior when it comes to Neanderthals. Well, there is a fountain of knowledge coming your way, and I hope you enjoy these conversations as much as I did. I know I always say that, but I really mean it. Every time when I'm sitting down with a guest, I'm so excited and I learn so many new things, and I really hope you do enjoy this episode. And with that, I'll leave you to the episode, and as always, stay curious, stay engaged, and welcome to the HEAS podcast. Hi, Mike, and welcome to the HEAS podcast.
Thank you very much indeed. Nice to be here.
Who are you and what is your job?
So, my name is Mike D. I lead the radiocarbon dating facility of the University of Groningen in the Netherlands. I do a lot of research on all different aspects of radiocarbon production, and then radiocarbon dating.
So your field of expertise is radiocarbon production as well as dating, which is good because we've covered radiocarbon dating in a previous episode. It was episode 4, actually. And although we might know a little bit about it already, could you quickly tell us more about what this method is and how it works?
Yeah, of course. So radiocarbon dating is a method of scientific dating that employs the extremely rare isotope radiocarbon. So carbon comes in essentially 3 natural isotopes: carbon-12, carbon-13, and carbon-14. And carbon-14 is extraordinarily rare. It's about 1 in a trillionth as common as ordinary carbon-12. I heard a story once that if you took all the people in all the world and you shaved their heads and you put all that hair into one room, one of those strands of hair would be radiocarbon and all the rest of it would be carbon-12 and carbon-13. That's how rare it is, so it's amazing we can actually measure it and get ratios of radiocarbon to carbon-12. That's what we do. So what happens is radiocarbon is produced in the atmosphere, and then it's oxidized to CO2. And the CO2 is taken up by plants through photosynthesis, And from there, it ends up in all living things because animals eat plants or they eat other animals. So this isotope of carbon, this extremely rare isotope of carbon that's produced in the atmosphere, ends up in all living things whilst they're alive. Now, it's also radioactive, obviously because it's in such low quantities, it's not dangerous at all. But when you die or when a tree is felled, for example, the proportion of radiocarbon relative to stable carbon in the biological compounds of that former organism, the radiocarbon decays away. So the ratio between radiocarbon and stable carbon-12 declines over time. So we have leftover remains from, say, for example, bones from the Paleolithic 30, 40, 50,000 years ago, and they have very little radiocarbon left in them. And how radiocarbon dating works is that we basically try to work out, because we know the decay rate of radiocarbon, how long ago it was that radiocarbon, that organism was alive, and that organism had the proportion of radiocarbon in it that would be consistent with a living organism.
Okay, you said C14 is radioactive. There is a follow-up question. I'm going to jump right into that because you've already mentioned it. If radiocarbon is radioactive, is it dangerous to absorb a lot of it during our lifespan?
No, in fact, it's not. Essentially, actually, the decay of radiocarbon is it's a weak emission anyway. So even if it were relatively concentrated, it wouldn't be terribly dangerous to living tissue. But because also radiocarbon, even in atmospheric concentrations, even in living tissue concentrations, is, it's at its most, it's still extremely rare. And so the dosage you would get, the radiation dose is so small, it's not at all dangerous.
As you mentioned in the beginning, that it's very rare. And which materials can be dated? I guess every living organism.
Well, it's interesting actually, because even more than that, really. So what you have is this constant production in the atmosphere, and organisms, plants essentially, absorbing the radiocarbon from the atmosphere. So all of those, all living species can ultimately be dated. You have to be very careful what compounds you select from those organisms, the remnants of those organisms, but they, in principle, can be dated. But also, interestingly, you can date other things too. Like, for example, the hardening of various concretes involves the absorption of CO2 from the atmosphere and the building in of that CO2 into the chemical structure of the concrete or the mortar and this sort of thing. And if it's locked in there chemically, then the only way it's really ever lost is through radioactive decay. So you can take some inanimate material that has once upon a time been in equilibrium with the atmosphere and absorbed a certain amount of radiocarbon, but then a period of time has passed and it's lost radiocarbon through decay. You can work out how long it was since that was a liquid slurry of material setting and absorbing radiocarbon from the atmosphere. So even beyond living organisms, one can date other things as well.
That's a pretty useful method then. And I've read in your publication record, there was mentioned that there is a tree ring radiocarbon record. Can you explain what that is?
Yeah, so this is important because it's quite hard to explain, but the production of radiocarbon in the atmosphere is not always exactly constant from year to year. I mean, the method would be very simple if it was always unchanged. But what happens is that actually the starting rate, the production of radiocarbon in the atmosphere, varies from year to year to year. So what we needed in order to correct our radiocarbon analyses, when we're getting our radiocarbon to stable carbon ratios, was we needed to know what the original concentrations in the atmosphere were back through time. And one of the best ways of doing that, of course, is to go to tree rings. So tree rings what they do is they absorb radiocarbon in the year that they're growing, and they lock it in their structures, their chemical structures. And one of the most stable of those chemical structures is cellulose. And what happens is that the carbon that's in that cellulose doesn't exchange with the ring before or the ring afterwards. Indeed, it records the radiocarbon ratio from the year in which it grows. Now, you have these long, they're called dendrochronological archives of wood. We have very long bits of wood with varying patterns of the width, essentially, of the tree rings, and you can match them up from tree to tree in a given location for a given species. And you can build these very long continua of tree rings, and even you can dig up trees that have died long ago and were buried in swamps and so on, and they haven't decayed away, and you can match their ring width patterns as well. So we do have these archives, these dendrochronological archives of wood, where you can go back to them and say, I want a tree ring from the year 5469 BC, and they'll give you exactly that tree ring because of the sequence they've built up. And so what we've done is measure the radiocarbon concentration in those tree rings back through time. And our tree ring record now goes back, 14,000 years or so. And that, by comparing that with what we would expect the radiocarbon in the atmosphere to have been in that year, now, having lost a certain amount due to the time that's passed, we can correct our dates, you see, for the absolute concentration of radiocarbon in the atmosphere at the time, because we now got this reference where we can measure it, because we know the age, the calendrical age of each of those tree rings in advance of doing the radiocarbon measurement.
And you mentioned that the concentration of C14 in the atmosphere was not always constant. Could we see a change with, for example, radioactive bombs that were tested or during war times?
Yeah, so this is really interesting and actually a big application of radiocarbon analysis. I like to distinguish between radiocarbon analysis and radiocarbon dating. So there's a lot of people who work in radiocarbon science who actually don't date anything. And a lot of them work on what's happened in the atmosphere in the last 50, 60 years. And that's because when there was overground nuclear bomb testing in the mid-20th century, mostly by the Americans and by the Russians, you got an excess production. Well, this is what we think anyway. Excess production of neutrons in the atmosphere, and they are the precursors in many ways of radiocarbon, the isotope radiocarbon. So you suddenly got an enormous increase in the concentration of radiocarbon in the atmosphere that could have never occurred naturally. So you have a big, what we call bomb spike of radiocarbon that reached its peak in about 1963 or so from all of these atmospheric nuclear bombs throwing out neutrons and producing radiocarbon. Now, there was a international treaty signed in, I think it was 1963, and from then on, atmospheric testing was banned. It still continued, but it continued underground. And so actually there wasn't anymore the sort of release of radiocarbon into the atmosphere that had been going on in the mid-20th century. And now you have a big decline in radiocarbon in the atmosphere from this huge peak in 1963. And you can see it drop back down to, it's just actually got down now below sort of normal levels after 70 years or so. Is it 70 years or 60 years? And that decline in the concentration is actually not due to radiocarbon decay. That's just to do with the slow absorption of radiocarbon, by the trees and so on through photosynthesis, but also into the ocean, the disappearance of it into the ocean. And that analysis of the variation and that pattern of how quickly it's disappearing into the ocean and into the plant life and so on is a big resource for people studying the carbon cycle. And especially now, I don't know if I just want to keep going on this, but I think it's an important point when we're burning a lot of fossil fuels, especially in the last sort of 50 years. Fossil fuels have been underground for a very long time. Oils, coal, this sort of thing, contain a lot of carbon, but it's all carbon-13 and carbon-12. the original radiocarbon that was part of the antecedent plants that have, died and become oil or other organisms, all of the radiocarbon has disappeared many, many years ago, because this is millions of years old. So when you burn coal and you burn oil, all you get is carbon-12 and carbon-13, the stable isotopes of carbon going into the atmosphere. And that process has decreased more rapidly than otherwise the concentration of radiocarbon in the atmosphere, the ratio of radiocarbon in the atmosphere. You have a expected ratio of carbon-14 to carbon-12 in the atmosphere, but if you start chucking in more and more carbon-12, you decrease that ratio more rapidly than otherwise naturally would have occurred. So this decline of the bomb curve and its loss in the atmosphere is related to the activity of the oceans and the plant life, but it's also a big source of data on how much fossil fuel burning has been going on. So there's lots and lots of research that can go on into that field.
There's actually many more applications than you would initially think when you read about C14 dating. As you said, there is two things, the analysis and the dating. Thanks for that explanation. You must be a very good teacher. I think we're taking a lot away from this conversation today.
Thank you.
Now, is there any specific project or maybe paper that you would like to talk about?
I guess the easiest one for me to talk about is the work we did on the first Europeans to get to the Americas. I guess that's the one that people want to hear about. I have to first start by saying a lot of this work is based on the original research by a young female Japanese researcher. She was only a postdoc at the time, I believe, called Miyake in Japan. And she discovered that at certain points in the past, by measuring actually radiocarbon in tree rings, there were incidences that occurred where the concentration of radiocarbon in the atmosphere spiked very suddenly. Spiked a bit like the bomb curve, not anywhere near the magnitude of the bomb curve, but spiked very suddenly. I could go into all the reasons for that, and maybe we'll get onto that, but this was a huge discovery, really. And it also meant that because she was measuring it in tree rings of known age, we knew exactly the years in which these spikes occurred. And so they became time markers. If you could find them in artifacts, in archaeological objects, particularly measuring tree rings and wooden objects, if you could find these spikes in radiocarbon, you would know exactly what year you were dealing with. And that was a game changer, because now we could use this method not only to estimate things to the nearest couple of centuries or so on. If we found this spike, one of these spikes of known age, all you needed to do was, that tree ring represents that year, count the rest of the tree rings out to when you reach the bark. That was when the tree was cut down, to the exact year. And it couldn't be any other year. So we put this to the test, really, a couple of years ago, when we got access to, through the help particularly of someone I should mention too, Birgitta Wallace, Dr. Birgitta Wallace of Parks, Canada. We got access to some wood that she had kept from the excavations of the Viking, the Norse layers in Newfoundland in Canada. And we were able to measure through the tree rings of some of this wood and find one of these known age spikes. And it turned out that by counting the rings in each bit of wood to the edge of the piece, that three different pieces that we analyzed from three different trees, the wood people could tell us that, all had exactly the same cutting year. And the chances of that randomly occurring on sort of driftwood or deadwood or whatever else is pretty much nil. It's not nil, but it's very low. And when you've got it from the Norse layers with cut marks in it by Norse tools, we were able to argue that it was the year in which the Vikings or the Norse were in North America already cutting down trees. Now, this was actually relatively close to what the age people expected, but it was an exact year. And it was 1021. AD, if you like, which was exactly 1,000 years before we did it, which was kind of a coincidence. And this 1021 then represented, if you like, the first calendrical date for Europeans in the Americas before Columbus. It's sometimes misconstrued to be the year they got there, or, I mean, all we can tell is they were there at that time cutting wood. We don't know. You'd have to go through the entire archive, and you'd have to be lucky to find the spike and various other bits and so forth. And it would be quite tricky to prove that they were there earlier or how long they were there and so on. But as I say, out of a random sample of ones that we took, the chance of getting exactly the same year on all of them was so rare, so unlikely, that we took it as pretty strong evidence that we had a fixed date now, the first fixed date for when Europeans were in the Americas, and indeed the Atlantic Ocean had been crossed. So that was quite a big paper, and all of it really stems back to the work that Fusumiyaki did on the discovery of these sudden spikes in radiocarbon production in specific years in the past.
That is really fascinating and such a cool study, and really remarkable.
Thanks.
That you can identify this peak actually and then like calculate back, or like in whichever direction you want to go. That's fascinating. Thanks for telling that. And another question that I actually wanted to ask before that, but then forgot because I'm never taking any notes. In research papers, sometimes you see, and it could be confusing, calibrated and uncalibrated dates. Could you explain that?
Yeah, that's a very good question. So it is tricky to explain, a straightforward way, but I'll try. So calibration in its general sense, of course, just means correction or obtaining the right result from the numbers you have, making sure that it's correct. In the beginning, when radiocarbon dating was invented, there was an assumption made, which I think even the earliest radiocarbon scientists knew was probably incorrect, but it was what they were working with. And that was that radiocarbon production in the atmosphere was constant back through time. And if that were the case, then actually you wouldn't need to calibrate dates as such. The result that you get, which is the ratio of radiocarbon to carbon-12, then some mathematical manipulations gives you what was called a radiocarbon date. A radiocarbon, it's an arbitrary scale really, but it's a radiocarbon result that was called a radiocarbon date. Now, when we discovered that radiocarbon concentrations in the atmosphere were changing or had changed through the past, sometimes quite dramatically, especially when you go back into the ice ages and so on, then those numbers we were getting that we were calling radiocarbon dates were not really dates at all. They were just numbers. So we invented a procedure called calibration to turn them into sort of any sort of date that we could recognize. And that effectively involves comparing the radiocarbon date with the historical record of radiocarbon concentrations in the atmosphere, mostly obtained from measurements on the tree rings of exactly known age, getting the result from that comparison, and that is an absolute time result, like a calendrical time result, what most people would know to be an age. And that process of correction of the original, still called a radiocarbon date, to a proper calendrical age is called calibration.
Thank you so much for finally explaining that. I mean, not finally explaining, but for me to finally understanding that. Let's move on to the second follow-up question, although you've already touched that question. Where does C14 come from?
Yeah, it's a good question. So radiocarbon has a half-life about 5,700 years or so.
5,730. I learned that in episode 4.
Well, then, this is one estimate actually. We don't know it's but anyway, yeah. It's called the Cambridge half-life, the one year, but that's true. So, because the Earth is millions of years old, it would all be gone if it had just been created at the beginning of the formation of the Earth and so on. It is being produced all the time in the atmosphere. So why is that? How is that? And what happens is that the Earth is constantly being bombarded by particles from space. In the beginning, they were thought to be waves, actually, electromagnetic radiation, but they're actually particles but we still call them cosmic rays. Again, it's a sort of an anachronism, a misnomer from the original time when it was thought to be waves. But they're particles, they're charged particles. They come from a lot of charged particles come from the sun, but the ones that are mostly involved in radiocarbon production come from elsewhere in the Milky Way and even outside our galaxy. And they come bashing into the Earth at great speed, and they smash into the nuclei of atoms in the atmosphere, nitrogen and oxygen and so forth. And what you get is it's called spallation, a splitting up, if you like, of those nuclei, and a rain actually called an air shower of subatomic particles. As all of these nuclei fragment and fall apart, you can see these air showers develop. All these subatomic particles come raining down towards Earth. And this is an ongoing process all the time. And in amongst all of that, the free neutrons that are released in much the same way as neutrons were released by the bomb, by the nuclear bombs and so on, the free neutrons that are naturally released by the cosmic ray bombardment, some of them get captured by other species, chemical species in the atmosphere, usually nitrogen, and that is how radiocarbon, this isotope, is formed. It's indeed why it's called a cosmogenic isotope, because it's the cosmic rays from outer space that smash up the nuclei, release these neutrons that get captured to form radiocarbon.
Now let's move on into the HEAS quick takes. I'm going to ask you a couple of rapid fire questions.
Okay, do my best.
All right, let's get started. Where are you from?
New Zealand. Am I supposed to speak fast too, or not?
No, it's fine.
Didn't know if you were going to go really quick. Yeah.
No, we still have time. We're still...
Okay. New Zealand.
Fine. Why did you become a scientist?
Oh, that's a good question. Crikey. Am I allowed to tell the truthful answer? Or am I allowed to Look, I guess I say that I was working in the city in London, in the financial area, and it was a great job in many ways. I wanted my life to be something different from the work I was doing there. I wanted to make this is a bit vain to say it like this, but I wanted to make a more meaningful contribution to things. I wanted to at least do something. I think I probably wasn't creative enough. I would have loved to have been a musician or something like that, and done something really nice that would have added to the human experience. And perhaps due to lack of talent on that front, I always had in the back of my mind that I had studied science at university, obviously, that I could go back to science. And I felt that my work in the city was not fulfilling me, I guess, in the way that I wanted to. And I really wanted to be a researcher, to create something new, to leave something, to, I guess, have a legacy. And so I ended up going back and doing a PhD in my 30s. Yeah.
That's not vain at all, in my opinion. What do you collect?
Oh, that's a question I often get asked. What do I collect? Yeah,
really?
Because I don't really collect things.
That's good. I wish I could say that.
Oh, really? Because, I'm not really much of a gatherer of things. I mean, I guess when I was young, but people often ask me what sort of hobbies I have, and I'm afraid to say most of them relate to work. So it doesn't really count as a hobby. No, I don't suppose I do collect things terribly much. But it's a perfectly valid question. I don't know if that was from the previous interviewee, was it? Or no?
No, I'm just interested because I asked that question before, and I got a lot of interesting answers to that.
Right.
But I guess not gathering or not being prone to gathering stuff makes for a clean house. I mean, I do not intentionally gather stuff, but sometimes, I need to, clean out the house again to get rid of stuff.
I think that is true. And in fact, I don't own all that much stuff. My house is pretty empty, really. I guess now, too, the way everything's electronic, one doesn't even sort of collect CDs. I'm now making myself old, or books even so much. One gets them electronically. So I have fewer and fewer things, I think, as I get older, which is probably the reverse of how things used to be. But anyway.
I would like that, but I do collect books. I don't like the Kindle.
Oh, you don't like them at all?
I don't like it.
Everything on my...
I'm having an iPad in my hands, but it's just for the fact I would love to have like a notebook or something, an actual one. Yeah. But if you turn pages, it's not good for the audio. So that's why I'm like...
No, true. Yeah.
All right. 3 words to describe yourself.
Okay, tried my best. I'll say that I am dedicated, I would say, to my work. Really, I am. I think I'm, I don't know. It's, again, quite a hard question to answer with and still remain modest. I think, I know I've got a good one. Impatient. I'm impatient, and that's something that's worked against me quite a lot being a scientist. I always want things done tomorrow. But on the other hand, it does turn me into a very sort of, I guess dedication is similar to the ability to work hard. And I think hardworking would be the other one. I don't suppose I'm naturally gifted in the way other people are, but I am dedicated, hardworking, and probably impatient. I think that's the three. They're all quite negative, really, but that's probably how I would describe myself. And perhaps other people would too. I don't think there's any escaping the fact that I do work really hard.
We should ask your PhD students that question.
I think, yeah, that's good.
Yeah. Are you an introvert or an extrovert?
Well, that's a good question, because actually, to contradict myself a little bit, it sounds like I'm locked up in a room working all the time, but I'd say I'm an extrovert as well. I'm not an introvert, which has helped me in academia, actually, because contrary to what a lot of people think, to be, I think, an effective academic, you have to stand up in front of an audience and perform all the time. It seems a bit funny to put it that way, but, in front of students, you've got to inspire them. You've got to lead them. You've got to, convince, funding bodies and all this sort of stuff with a bit of a show, with a bit of a bit of bravado. And I think that being an extrovert and being prepared to speak up and be noticed, I think actually has helped me in my career a little bit as well.
I love to hear that. And I don't know if I'm going to keep that part in, but I mean, I think you're a very smart person just from the way you're explaining things. And like, that's just my view of you right now. And I've read through your papers and stuff, but I do hear what you're saying. I also do think of myself as not the gifted person. But on the other hand, I mean, that's so self-deprecating. I always say about myself, I'm the personality hire, but I do.
But the way I see it is that
output. I'm like, what I mean?
What I have learned, and I think I would like to say this actually, it's a more general point. What I would say is that what I have learned in academia is that it does involve a whole range of skills. To get ahead in academia, you have to have lots of different abilities. I've met some extraordinarily capable people who can calculate things, or very sort of logic based and deep thinking, hardworking sort of calculators, if you like. But they've struggled in their careers. I've met other people who were all show and no knowledge and no underlying perhaps ability, and they've struggled as well. And, there's a full spectrum there, and one needs to have a balance of different sorts of abilities. And I think that that's actually quite a good thing. Another one is the ability to collaborate with other people, especially in the sciences. I would say you need to be the sort of person that other people want to work with, because a lot of the breakthroughs, all of the stuff I've been involved with, that's turned out to be any good, has been me with a bunch of other people working together to achieve something. And you just like in any other sphere of life, I think you only really want to hang out with or to work with people you're happy to be around, and that they are kind and considerate and good fun to collaborate with. And so those sorts of skills are extremely important, I think, too.
I'm getting closer and closer to the conclusion by talking with people that this is also a very valuable characteristic or character trait. So yeah, I'm happy about that. Last one of this segment. What do you want people to know about you?
Wow, these are really tricky questions.
I'm not good at my quick takes, actually. That's
no, but that is what I want people to know about me. I have to say that one. I'm not so bothered what people know about me as such. I hope that in terms of my work, that I've been, I think the first thing is that I've been a decent person to all those around me, to my students, to my PhDs and to my colleagues and so on. And that I that people know that about me. It is important to me because sometimes you hear about these early scientists, brilliant people and so on, but horrible individuals. And I don't think I'd want that legacy, you know. I first want to, I think, be known to be a decent person. But then, I'd like to think that in the future, I have done something that people will remember, or that they will cite, or that they will refer to now and again in the future. That's all I think I ask for.
Now let's move into the segment where one of our previous guests leaves a question for you. And the question is there any step in your academic career that you wish you had done differently? And if so, why?
Oh, that's a really good question too. I have to say, an academic career is a series of steps, and each one is very tricky. It's tricky to get a good PhD position. It's very tricky to go from a PhD to a postdoc, get a good postdoc position. Often you have to move somewhere away from where you've been living, maybe from your home country and so on. And then to go from one of your postdoc positions to a permanent job is also tricky. And just not so many years ago, I passed my tenure track to get a sort of faculty position in my university. That was also very hard. So they were all difficult. Would I have done any of them differently? I mean, if I had had my way, I guess I would have done fewer postdocs. I don't know. I mean, it would have been nicer to have ended up with a permanent job earlier, because I sort of came back to academia in my 30s, and I was already a bit older, and I was living in England, which was a very expensive place to live at the time, and I couldn't afford anything. So perhaps, I don't know if I could have controlled it though. If I'd had to make the transition from sort of first postdoc to a permanent position earlier, I would have preferred that. Maybe I should have applied for jobs in other countries outside of England earlier on. Maybe I would have been happier to have done that, because I would have been probably where I am now, but sort of 5 or 6 years earlier. That would have been nicer. Yeah.
But there's no such thing as a timeline, and everything you did came with its own experiences, and you've grown and learned from that. So maybe it's just the right time for that.
Yeah, and I look back and then I think I wouldn't have had those good final years I had in England. I met other people, and so it's hard to know. And what I do think about academic careers is that there's so little really that's under your control. You do your best, and quite often it's a sort of a lottery, or you put your name in a hat and you hope you get picked. So it's pretty hard to say I would have done things differently, because I don't know if I would have been picked if I'd have done things differently. So yeah.
Exactly. Where can people find you online?
Oh dear. So I was on Twitter when it was Twitter, but our project was, but now I'm no longer on it. I have a new website for my current project. I don't know if that has my email on it, but I have a website via my university that probably has my email on it, if people really wanted to find me. There they can see things about my career too, if they type my name into the net. I'm sure I work at the University of Groningen in the Netherlands, or probably in English it would be like Groningen. But so if people were desperate to find me, then that's where they would look, I think.
And Mike, to wrap this up, is there anything you would like to add to this conversation? Is there anything you feel like I didn't ask you?
No, not so much. I mean, there were very tricky questions, challenging questions, and I enjoyed it. So, thank you very much for your time.
I enjoyed it very much. Thank you so much for sitting down with us for the HEAS podcast.
Pleasure.
Welcome to the HEAS podcast, Mattias.
Thank you.
We're so happy to have you as a guest. But to start, could you tell our listeners who you are and what you do for a living?
All right. So I am Mattias Jakobsson. I'm a professor of genetics at Uppsala University. And as any university professor, I do a mixed bag of teaching, research, but also outreach, speaking to you and speaking to the public in general, especially when it comes to genetics and human evolution and human history. And those are also my topics when I do research. I'm really interested in the human past. both on a very deep time when we're going hundreds of thousands of years back in time and looking at the evolutionary changes, but also the more recent patterns of movement of people.
So to sum up, you are interested in population genetics and in human evolution, and you're the head of a lab at the University of Uppsala. Is that correct?
Yes, that's correct.
And can you tell us about the key questions that your lab is currently investigating?
Right. So there's a few key questions. One is that we're trying to go deep into time following the human lineage. And if you're following the human lineage, you can try to figure out what genetic changes happened on this lineage. Maybe we can use Neanderthals to compare with, and then we have a way of figuring out what's unique to us humans. So that's one trajectory in the lab. Another trajectory in the lab is to work with what has happened in the last, say, 10, 15,000 years in Europe, and as I spoke about here at the University of Vienna today, on North African patterns, typically then patterns that are related to migrations, so where new people have come, and often they're related to other phenomena like new ways of life.
As you mentioned, you gave a talk earlier today at our university. I was really fascinated by the Northern African part of the Neolithic story that you told us about today. Could you maybe sum that up quickly for us here?
Yes. So we know reasonably much about the Neolithic process in Europe, but Northwest Africa is or has been an enigma. There are two different competing theories, and there are signs supporting both. So you can imagine that there's a group of people living in Northwest Africa. They're living as foragers, hunting and gathering. And there's a possibility that they invent the farming way of life. They start domesticating crops and animals. That's one scenario. An alternative scenario is that you actually have people from Europe, because at this time point, there are people in the south of Spain that are living as farmers, that they actually jump over or sail over or however they cross the Gibraltar. And in that case, then you have another group of people coming in, bringing this new way of life into North Africa. And that was really the study and the talk today. And what we could see is that, yes, there is clear evidence of people migrating from the Iberian continent just across to the other side into Morocco, today's Morocco. And they're genetically very similar to the farmers who are living on the other side. But they've also mixed with the local groups who live in North Africa. And then there's also interesting signs that other groups in North Africa start perhaps being inspired by this new way of life and start testing with local plants and animals to see if that way of life works for them.
So the classical theory is that farmers came in from the Anatolian side. Is that correct?
Yes.
And then took the coastal route to the Iberian Peninsula. And then you said they could have jumped over, I'm doing quote unquote. And then in the ancestry, you can see that they mixed with the local people from Northern Africa. So what's the possibility here? Could it be that the Northern African people could have invented this way of life before that, and then still the people from the Iberian Peninsula jumped over and they still mixed? Or do you think that this way of life came with the Europeans? It's
certainly possible, the way you describe it, that it was invented first in North Africa. But currently, the archaeological record points the other way around, because the earliest dates are from those archaeological sites with the remains that are genetically similar to the people from Europe. So if there are other sites that we investigate that have the local ancestry of the people in the area, and also indications of domesticated animals and plants, maybe we're pushing that time back. So maybe it's a competing, going further and further back in time. And then we might change our interpretation again. But at this stage, the archaeological record points the other direction.
So it coincides with the time of the arrival from the people from Europe.
It does.
And when was that?
This we're talking about approximately 7,000 years before now.
Okay. So that also means that still farming was invented before that. And it could have happened in parallel in Northern Africa, but the current state-of-the-art evidence points towards that it came with the Europeans.
Yes.
All right. Thank you for clarifying. That's so interesting. I've read that your lab uses computational approaches to investigate the human genetic variation and to understand human evolution. What does that mean? Does that mean you're not going to the field yourself? I mean, obviously, you mentioned you're not an archaeologist, but people from your team. So you don't dig in the field, you get probably the data. And then could you walk us through the methods that you're using?
Right. So usually these projects are big collaborative efforts. So sometimes we have archaeologists actually working within the group in Uppsala. So we have a number of employed people who are archaeologists by training. But sometimes it's collaborative efforts with people who have done excavations. And typically then you start discussing with the people who have excavated a site for a long time, and they're interested in seeing if something else can be added to this understanding of the site. And genetics is, as we've learned in the last 10, 15 years, a great tool, and with the Nobel Prize kind of showing that it's not impossible to get it from very old remains, human remains. This is a really, really exciting tool. So then we get going on that, and then typically there's a sampling period, and sometimes I don't do the work myself, but I usually travel with people, and there's quite a lot of effort with making sure that everyone is comfortable with what's being done with the material, and also interacting with the people who might be really, really good experts on those specific sites. So we learn from each other, and that usually takes multi-years, we're talking here. And then there's a long process of actually getting DNA from the human remains. You sequence the entire genomes. It's 3 billion base pairs in the best case. And then we come to the computational part. So then you have a very nice, hopefully from many individuals, genome. And then you can compare variants in that genome to modern day groups, but even better to contemporary groups with whichever site and time period you're working with. And there is a very fundamental genetic understanding, and that is that if an individual carries the same genetic variant on a specific site, then at least at that specific site, they're slightly more related to each other than they are to people who carry an alternative variant at that site. And the beauty of the genome is that it's very, very large. And then there's a phenomena called recombination that kind of mixes up this genome. So that allows us to do very good statistics on such patterns. And here comes sort of the computational part. And you're working with information that might be many million variants large in one direction, and perhaps maybe many hundreds or thousands of individuals. So these are large data sets that you need to handle with computational tools.
And now that you computationally found out about the differences or similarities in those genomes, you probably have to visualize that because you're working with a lot of populations and a lot of groups, right?
Yes. So there's really two very important components. One is the statistics, to making sure what you're seeing is actually statistically sound. And the other, as you say, is visualizing it, because it's quite easy to compute a statistic and get a p-value, but that's quite difficult to visualize and get other people to see what you are also seeing in the data. So then visualization comes into play, and that's an extremely important part for science, but also for bringing science to the society in general.
For our listeners, I'm going to put in a PCA, which is one of the main visualization tools I think we use in genetics, so that they can see what we're dealing with. And now that you walked us through the methods a bit, and we learned more about that, what are the challenges you face in studying ancient human populations?
Well, one of the challenges is that there are not that many excavations and remains, and that is an increasing challenge the further back in time you go. So there were fewer people if we go 1,000, 10,000, 100,000 years back in time. So there are simply fewer remains left of those individuals, but also sites where they've been living. So that's a challenge. And also the longer time goes, more recent human activity may destroy these sites. So that's a challenge. Another challenge is to actually get DNA from whatever material you're working with. And that's also something that's important for preservation for the future. So if you sequence the entire genome of a human remain, you will never have to go back and sample that specific individual again because the genome is sequenced. Mm-hmm. So there is a sort of an element of curation of the material and also preservation and making the data available to anyone.
All right, let's move on to the next segment, which is the follow-up questions. So as I told you, I go on Instagram and our listeners have the opportunity to ask our upcoming guests a question. And that's the one that was left for you. When you find yourself stuck and hitting a dead end at work with the lack of ideas and motivation, what do you do to clear your head and pull yourself out of it?
Ooh, that's a good question.
I agree. I liked it.
Right. Well, usually, I'm often more faced with the opposite of that phenomena, where I have a lot of nice and good ideas that I want to pursue, but there are so many other things that comes with a job that you have to deal with. So the amount of time you can spend on research is still limited. But otherwise, I think I'm quite often thinking very clearly when I'm in a public place. So I go to coffee shops and sit and work, and I like the buzz around me. I'm not at all disturbed by sound and people moving and talking. I'm quite productive in such environments, and that's a way. And when I need to write a grant or something like that, and I really need to focus, then I go to busy places than to sort of silent places.
That might sound so counterintuitive for a lot of people, but I do fully agree. I do the same. It's like the most productive time ever. I have a little coffee shop around the corner from where I live, and I'm frequenting them. They probably think that's my second home.
Right. No, and I think it's different to different people. Some need, calm and quiet, but at the same time, at the lab or the corridor where I work, when it's very busy and loud and people are engaged in talking, that doesn't disturb me, but perhaps others are disturbed. But that's sort of more inspiring, I think.
All right, now let's move on to the rapid fire section.
Yes.
Just try to answer quickly. If you want to elaborate on something, please go ahead.
Sure.
Where's your next trip going?
My next trip, it's going home. But after that, I'm going to the Swedish mountains to do some skiing in a few weeks.
Really?
Yeah.
That's amazing. Do you still have probably a lot more snow there than we currently have here, because we're struggling with the snow in Austria?
Yeah, it's on and off. So, this winter has been quite good in the Stockholm-Uppsala area. We've had almost half a meter of snow, but most of it has melted away in sort of this central part of Sweden. But in the north, there's lots of snow.
Yeah, I bet. I'd love to do that too. What was your least favorite subject at school and why?
Swedish. Like Swedish language. I'm not very good at spelling and grammar and these types of items.
If you weren't a scientist, what would you be?
Well, I've had different thoughts on that during my life. And at one point I wanted to be more of kind of a person who are out in the forests. But I realized, for some reasons that didn't attract me at some other point. I might be a lawyer. I think that I like these sort of formal rules. Like genetics is attractive in the sense that there are Mendelian laws and rules. It's a bit like mathematics, and I can see that perhaps law, but that's maybe my very confused and unexperienced view of how legal system works. But I think that there are strict rules and how to work. That could be interesting.
I could see you as a lawyer.
Okay, that's interesting.
Tea or coffee?
Coffee.
As we saw. And what do you want people to know about you?
Oh, wow. I often get the question of, have you done an ancestry test yourself or something like that? But quite often the answer to that is that, well, my parents have done a bit of genealogy and they've figured out that, okay, my ancestors have been living in the southern part of Skåne for quite a number of generations. So it becomes rather uninteresting to do it for me. So then you would have to think about other things. Yeah, what's interesting? I had an alpinist career when I was younger. That was kind of a lot of fun. Went to a lot of fun mountains in the Himalayas in the US and various places of the world.
Oh, wow.
Yes, I'm a very retired alpinist at these times.
But that also kind of comes full circle back to when you said you wanted to do something in the mountains and you're going to ski. So yeah. All right. Then the next segment is called Speakers Ask Speakers. And one of our previous guests has left a question for you, which is, which equipment would you buy and for what if you had no funding limit? As you're working computationally, that probably doesn't apply to you as much as for like people that have to buy, lab equipment and devices that could easily exceed a couple of €1,000.
Right.
But what would you get for your lab?
Well, that would be an easy question. I would get a very large computer cluster with very large integral storage. We have a very, very good facility in Uppsala and sort of a national facility in Sweden. But just the other month, I had one of my colleagues used up 100,000 computer hours in a month. And that meant that we were kind of at the limit of what we were allowed to use. And now we need a lot of computer power always. So that would be my purchase. And it would probably come with a technician, because just buying the computer or a large computer cluster, that's very nice and it's going to work for a month. Obviously. But very rapidly, you're going to run into trouble unless you have staff.
That's a very good answer. Now to wrap this up, Mattias, where can people find you online?
Well, the easiest, I'm kind of an active social media person. I stay away from those phenomena. So it's the website, Uppsala University. There's a lab website.
And is there anything you would like to add? Anything you'd like to announce? Upcoming papers or anything you feel like I didn't ask you?
I can point to a very interesting paper, I think, that we published on Friday. It's a small team of people. It was a review paper in Cell, but there's embedded in that paper a nice potential model for the genetic making of modern humans. And we'll see how that will be received.
All right. I'll put that paper into the show notes below so our listeners can take a look at that.
Thank you.
Mattias, thank you so much for sitting down with us for the HEAS podcast. It was a pleasure.
Thank you.
Hello, Wil.
Hello.
And welcome to the HEAS podcast.
Thank you. It's a pleasure to be here.
We're thrilled to have you on. And to kick this off, could you please introduce yourself to our listeners and tell them what your job is?
I'm an archaeologist. I work at Leiden University, where I am professor of Paleolithic archaeology. That is until 3 months ago, because I'm officially retired now, but still kicking.
Still kicking,
yeah.
As we could see from the talk you gave earlier today. So you're an archaeologist with a lot of experience. Looking back at everything you achieved, can you talk about one project that was specifically outstanding to you?
Ooh, that's a difficult one. I think that most colleagues would, if they were asked such a question, they would probably say the same thing as I'm going to say now. That's the stuff that I'm still involved in. So that has not been published yet. The material or the issues that I discussed this morning in my talk, that's fascinating, and I'm pretty proud about the project that we have and the team that we have. So with something like 30, 40 collaborators from different disciplines, various disciplines. And that's what makes this topic, this subject, this field so exciting. That when I was a student, you had earth sciences, ethnography, cultural anthropology for comparative exercises, and archaeology. And now you have zillions of methods, and people from all kinds of disciplines that help to understand how we became such a strange primate that we're now sitting around these equipments recording a podcast.
You're so right. It's actually weird if you think about it. So you mentioned this whole network of various disciplines that if they come together, I think we can do great science.
Yeah.
And this project where so many collaborators are involved, I know you've mentioned it in the talk, but people who are listening to this podcast probably didn't listen to the talk, which you can listen to and watch on YouTube, by the way, for our listeners. Do you want to give us a quick overview on this project?
Well, the research it is about last interglacial, that is a warm temperate period between 125 and 115,000 years ago, with a climate more or less comparable to the one in Europe nowadays. It's a last interglacial ecosystem focused on lakes near what is present-day Halle in the former German Democratic Republic. There were huge brown coal mines there, and those brown coal mines that destroyed the landscape, but at the same time allowed archaeologists to discover very important archaeological sites. So it's what we call discovery through destruction. And in the 1980s and 1990s, there was a very active team of archaeologists headed by Dietrich Mania, a professor from Jena, and they salvaged in front of these huge quarrying machines hundreds of animals that lived there 125,000 years ago, the remains of campsites of Neanderthals who were hunting those animals, exploiting those animals, butchering those animals. And when the mines, those huge brown coal mines, we're talking about thousands of hectares large and more than 100 meters deep, when they were shut down because they were ecologically pretty problematic, to put it mildly, though that landscape had to be reclaimed. And in those reclamation works, other archaeologists, including me, came in to excavate a few of the remaining sites that Dietrich Mania could not work on in the 1980s and the 1990s. Yeah, and we discovered something like a last interglacial zoo there with enormous amounts of animals, charcoal, remains of campsites of Neanderthals. We basically had 5 years' time to excavate very systematically a tiny part of a landscape that Dietrich Mania had to study in rescue archaeology. We had the means to take the time to study this very systematically. So we got a very high resolution record that we could connect to the humongous amount of material that Dietrich Mania was able to salvage in the 1980s, 1990s. And that material allows us to suggest that Neanderthals, who lived there for a few thousand years, were there all year round, hunted the largest animals of the Pleistocene, elephants up to 4 meters in shoulder height and weighing up to 13 tons, which must have created enormous amounts of food for these people. And their activities there probably also entailed modifying the landscape and suppressing forest vegetation and keeping the landscape open. So that is, in a nutshell, what we are doing there.
That's really fascinating. I actually have a bunch of questions. You mentioned Neanderthals. Did you find Neanderthal remains as well, or just what they left behind, like their probably cut marks on bones?
That's a good question. I shouldn't have used. I think to be precise, I shouldn't have said Neanderthals, because we've been fooled so many times about assuming that species X or species Y produced this record. But in that time period, 125,000 years ago, in Europe, there is thus far, I have to say, no other hominin represented than Neanderthals. But no, we didn't find any Neanderthal bones at that site. What we did find are stone tools, cut marks, bones, heated lithics and stuff like that, but no physical remains of Neanderthals. So I should have said, I hope I said that this morning in the lecture, hominins rather than Neanderthals.
You did.
Okay.
One of your research focuses is also Neanderthal studies. And that brings us back to 12 years to the past. And this is a question that I'd like to ask you because I'm very interested in that. So from my perspective, I have to say, I was intrigued to read your paper about early ochre use in Neanderthals. And to give a little context, in 2012, you published this paper on the earliest evidence of the use of red ochre by early Neanderthals that were living about 200 to 250 years ago. Now, ochre is known to be used in burial contexts, etc. And can you tell us more about your findings and why this is so spectacular when it comes to Neanderthals?
Yeah, that's a long time ago. This resulted from excavations that I started when I was still basically a student. In the early 1980s in Maastricht, Belvedere, that's in the southern part of the Netherlands. And it was my first excavation on my own, so I was very unsure, and I documented everything.
Yeah, that's good.
And we worked very, very slowly in those days. And I think that so everything that was unexpected, documenting everything that was unexpected, together with the fact that the sediments in which we were working were very bright, were very light, and not that much brownified, or how do you say that? There was no brownification through soil formation because the sediments were pretty deep in the interglacial soil that developed around 200,000 years ago. So it had escaped changes in color. So the small red dots that we discovered were visible. Had the sediment been darker, I don't think we would have seen it. And had we been much more, let's say, advanced in our studies, the people who were working there, we even might have said, yeah, no, forget about it. So anyway, we documented these dots, kept them, and then years later, we only started to analyze the material. And then it turned out to be hematite, and the hematite was not present in the background of the background sediments of that environment. We did experiments to reproduce the type of concentrations that we found, because we didn't find pieces of hematite. We found something like a film of hematite that enveloped the grains of the sediment. So in order to reproduce that type of work, we got in touch with a micromorphologist and we made thin sections of those tiny concentrations and suggested that these red dots were created when liquid in which hematite was solved fell on the surface. So we didn't find physical pieces of hematite. We found concentrations of grains of sediments that were enveloped in hematite. And they were not related to soil formation. We could test that with micromorphological studies by the late Hermann Mücher, etc. So, yeah, that was it. But then what does this mean? Exactly. Again, we have no clue. In the archaeological record, a lot is made out of the presence of hematite or red ochre, because, as you mentioned, it is often used in the later record in terms of it is often present in burial situations. But there are also, as we know from the ethnographic record, a lot of more profane types of use for that material. We have no clue how Neanderthals used it. We only know that they must have found that hematite probably to the south of Maastricht Belvedere in the Ardennes area where there are natural sources. They somehow turned it into a liquid that was dropped on the surface of that site that we excavated, and 250,000 years later we picked it up and were puzzled by the presence.
Yeah, that's spectacular. So the ochre in a liquid, if you think about it, that closely resembles paint nowadays. So you could draw that conclusion. Of course, as you mentioned, we do not know, but that's a possibility.
Yeah.
It's really fascinating to me because for a long, long time, Neanderthals were looked at these simple creatures, you know, these basic creatures, more ape-like than human-like. And if you think about that, they developed this artistical, ritualistic behavior at that time around. I mean, I think nowadays a lot speaks against that narrative already. But I'd like to, you know, always remind myself that this goes so far back, and it's not only modern humans.
No, it's also bearded vultures. You know, there are these, these vultures. There are bearded vultures that are documented to envelop their feathers in ochre-rich red water that stains their feathers, so they become red.
No, I've not heard about that.
Yeah. You look at
that
bearded vulture, ochre, and then the question becomes again. That's biologists ask the question: why are they doing this?
Is that for mating purposes?
That is one. That is one interpretation, that it is to attract partners or whatever. Another is that it helps to get rid of insects or pests or whatever. Right. So that is more or less medicinal. So there are, again, like with Neanderthals, there are also for these bearded vultures, there are various hypotheses why they do this, why they cover their chest and their neck in red material. So
that's so interesting.
Yeah.
So it extends way beyond hominins. That's fascinating. Thanks for telling that. I have to look that up. I'll put a picture in for our listeners because I definitely want to see that. So earlier this year, a new paper came out that identified modern human remains at a cave in Germany, which taught us that early modern humans had arrived to northwestern Europe by 45,000 years ago. Now that's much earlier than previously thought. That extends the time period that Neanderthals and modern humans have overlapped by a couple of thousand years, I think. And do you think, A, this timeline will be pushed further back with new evidence? And what does that overlap, you know, that coexistence of two hominin species in the same geographical region imply?
Yeah, that's an interesting question. I liked... Svante Pääbo, I think he talks about metapopulation, and in which there are different groups of hominins who are related. So I don't know whether the Ranis folks, the modern humans, would have been so different, whether they would have identified themselves as being different from the natives that were living in Europe. I don't know.
Yeah, you're right. What did they think about it? And they obviously mated.
Yes.
I mean, do you say that? I don't even know if that's the correct word to use as we're talking about humans, quote unquote. Yeah, that's a good question.
Yeah.
And that brings us to one of our follow-up questions that's, again, related to this topic. Do you think Neanderthals should be defined as a separate species from Homo sapiens? I have all the tough questions for you today.
Well, from a biological point of view, no, because they mated and created fertile offspring. So
that speaks
in that
against
sense, they're
the definition of
species. Yeah,
exactly.
Yeah, I think we archaeologists, especially the older ones like me, still have to come to grips with you're brought up with a specific type of stereotypes, and some of which we are very much aware of and others we are not. So, and stereotypes can be useful, but I think that the last 10, 20 years of especially genetic research has shown that we shouldn't take those words like Neanderthals or modern humans too serious. I know, of course, if you put skulls of a Neanderthal and a modern human on a table, even 4-year-old children will be able to discriminate, to identify. Well, they see that these things were different, but what that meant in terms of behavior,
Yeah, we can
I don't
only
know.
apply.
And we are often comparing the record of Neanderthals who existed for 300,000 years in Europe with the record of modern humans in Europe who arrived here 45, 50,000 years ago. So it's also comparing apples and pears to some degree.
And also, if you look at modern human populations, there is variation when it comes to, for example, tooth morphology and other structures. So there is that.
Yeah.
And we would also be able to discern the two or the variable groups. So it's hard to answer.
I think most of my career, I've been very ambiguous in this field. When I talk to colleagues who treat Neanderthals like, let's say, almost modern humans, like you have a smartphone and then there's a new version, it's just slightly different. I tend to emphasize the differences. And when I talk to people who emphasize the differences, I tend to take the other point of view. So it's because I think you need both points of view to make progress and to establish where research is needed to solve those ambiguities.
Moving on to our lightning round.
Lightning round, okay.
Yes. I'm going to ask you short questions and you can answer with a spontaneous thought, or you can also elaborate if you want. What's a hobby of yours?
I have, I like to walk, I like to hike when I can, out in nature, and an academic passion is art of Oceania, so New Guinea, Australia, the art that people produced in those areas. So that's why I'm going to the Wild Museum this afternoon.
Oh, right. You requested to go there. Yeah. It's nice. I hope you're going to have a nice afternoon. Are you a morning person or a nighttime person?
I'm a nighttime person, yeah.
I feel like I said that before in this podcast. I'm so repetitive, but so many researchers are.
Yeah.
They also like to work at night. What was your most exciting field trip and why?
I think the most exciting field trip was when we, in early 2000, working in near Perm in the Urals in Russia. It was very exciting because of I got to know a bit the Russian countryside, which had collapsed after the collapse of the Soviet Union infrastructure, and the beautiful landscapes and the people there, and also the desolate villages where beautiful wooden churches collapsing, and still a few, usually older females hanging around, keeping nice gardens and giving us the occasional vegetable and cucumbers to lighten up our very basic food that we had there and the archaeology. It's a beautiful area, especially for the early Upper Paleolithic. This was a few 100 kilometers northeast of the city of Perm. That was a very impressive fieldwork because of all the challenges, the alcoholism, I could go on for a long time about that fieldwork.
The traveling aspect of this job is really something special that not everybody can say of their own job. A movie or a book on an evening off?
Depends on the movie and the book. My wife is very much into movies and books, and I think I'm more into books. So.
One piece of advice you'd like to leave here for us?
To what I advised my students, so the first years and the second years and the third years, do whatever you like, but read as wide as possible, also in terms of methods. Read genetics for dummies, isotopes for dummies, dating for dummies. Try to get a grip of all the methods that are around. You'll never be able to understand them all, but it'll allow you to generate questions that other people might be able to answer. I'm a big fan of genetics for dummies and those types of books, because I think in order to oversee the field that we are into nowadays,
you need to be an amateur in all domains. I am not a special. I don't have any specialty. My specialty is that I don't have a specialty. I guess that I try to have a limited overview of all the methods and the approaches that are available. And I work with specialists, but I'm not a specialist. And for me, that's the most rewarding thing, because you're an amateur in both senses of the word, so that you don't know a lot, but that you also like that you also like the stuff that you're engaging with.
Well, you have no idea how reassuring that advice is and how comforting, because many people, especially during their PhD that I know, have this problem currently, me included, that we feel like we're not specialists in anything, but we scratched the surface of so many methods and so many things you could apply. But in a way, then sometimes we feel lost because, we're like trying everything, applying methods for certain projects, but we're not specialists in that sense. A friend of mine, she's done isotope analysis. She's also done the whole process herself. She wrote a whole paper about that. She also applied bioarchaeology to the samples she had. I'm talking about skeletal remains and genetics. So she did everything. And she just recently told me, I feel like this paper, I don't know if it's that good because I scratched the surface of everything and yet I'm not a specialist.
Yeah.
And we're happy that you just left this advice for us. It's really great to hear it from somebody like you who's so successful and has done such good research. So thanks for that. To wrap this up, where can people find you online, or would you want to be left alone in your retirement now?
No, I'm still working, so they can find me through my email.
Okay. I'll put it in the show notes below. And is there anything you would like to add to this conversation? Anything you'd like to promote? Or do you feel like I haven't asked you something that you would like to put out there?
I think I think the kind of collaboration that you see here in HEAS, so where you have people from all kinds of fields creating a new field, I think that's the future. But that's of course what's done at Max Planck Institutes in Leipzig, in Jena, and a lot of others. But this has a very fresh feeling. It's a fresh building. It's a beautiful building, and I'm a bit envious about what's going on here. So it was great to visit.
I'm happy to hear that. And thanks for chatting with me for the HEAS podcast.
Pleasure. Thank you.
Hello again, Victoria here. If you enjoyed this episode or the podcast in general, please consider showing us your support by liking, sharing, and subscribing to our podcast on Spotify and YouTube. Your feedback and engagement mean the world to us and keep us going. Thank you for joining us on this journey, and stay tuned for the next episode of the HEAS podcast.